So please unmute your mic.
Hello,
Yes, sir, you're audible now.
where are you stopped?
So go to the PowerPoint. Nothing. Everything is fine at your end. Good afternoon and welcome all. Today our esteemed guest is Dr. Selva Kumar. Sir is going to deliver the lecture on high-tech production and recent genetic improvement in tropical carrot. Sir, you are audible. Your presentation is visible. We welcome you on behalf of dignitaries, on behalf of NADCL, and on behalf of participants. So over to you.
Yeah. Good afternoon, everyone. Is it visible? Am I audible?
Yes, sir. Yes, sir. Yes, sir.
Yeah. Actually, I'm going to talk about high-tech production and recent genetic improvement in tropical carrot. So, how the carrot is evolved and what are the different types of carrots are available, and how the breedings will be done in carrot, and what are the recent genetic improvement, especially on quality aspects. If you take a carrot, it is a very typical root crop. Is a biennial crop and it is a unique crop which is evolved from the Middle East country. And if you see that among the vegetables, the carrot is a unique crop that initially that what we are seeing in the slides, that is it's exactly look like carrot. You know, are you able to identify which vegetable it is? Anyone?
We have turned the microphones off.
Hello?
So we have turned the microphones off. Hello.
Yeah, yeah. I mean, haha.
So we will, as we have turned the microphones off, they are not able to answer.
Okay, 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 okay.
So we
Sorry.
will turn them on at the end. So please ask the questions
Ah,
at the
okay,
end.
okay. Then I will continue. Okay. you know that whatever the crops which I am showing right now, this is a parsnip. This is a parsnip. It is exactly look like a carrot which are naturally cultivated particularly in the Iran, Iraq, and Afghanistan and Egypt regions. Only this is they initially they consider a carrot, but due course of time they it is a devoid of any colors, and it is look like white colors and dirt white color, and the name was they changed into a parsnip, because the carrot which was exactly looked like similar, because of the rich source of carotenoids, the name was kept in for the carrot as a carrot, and parsnip they have kept in the carrot initially they kept for parsnip, but they changed into parsnip. And if you see that in the diagrams, and what are the the excavations, what are the remnant parts are there in the Egyptian pyramids and Iran, Iraq excavation sites, they find that carrot and parsnip roots which are drawn in the wall painting, particularly in the Roman tower and particularly in the Ostia in the second century. And even if you see that some of the countries, especially in the Middle East countries, the orange and red carrots, the description was there in the 11th century. That 1950s, that Otta Banga, you might be known this very famous agronomist who are working in Dutch, and he studied about the complete carrots, the different type of color carrots which are lying in the European regions, and he studied and he collected a lot of different shape of the carrots, and he collected and he kept in the fans. And if you see here in these pictures also, that wherever the made demarcations, you see here that yellow carrots and white color mulli or radish they kept in the descriptions. That initially that whatever the carrots which are there in the Middle East countries, that is very typical mustard yellow colors carrots only which are there in the Middle East countries. There is no sign of orange carrots in the Middle East countries. You can see in these pictures also, the 1567 also they have find only the orange and black, means especially that yellow and black carrots which are there in the in their Middle East countries. So this what are the whatever the paintings, what are the descriptions, illustrations which are there in the Middle East countries or Afghanistan, showing that the black carrots and yellow carrots, which are completely belongs to Middle East countries, is because the carrot was originally is evolved from the foothills of the Caucasus regions or Middle East countries. Those carrots originally the black carrots and yellow carrots. And these carrots, the black carrots and yellow carrots, they once the Europeans, once they came for the Middle East countries, they have collected the black carrots as well as these yellow carrots. They took to the European countries and they have introduced those carrots. And those Europeans, they came to Middle East countries for searching the fertility enhancement for purpose. They want to identify some of the herbs and medicinal crops. They have collected the fenugreek and fennel and coriander and parsley. Along with that, they have collected the seeds of the wild seeds of carrots, that is a queen anise lace, which was does not have any type of colored roots. Particularly it was showing the branched carrots or white carrots they have collected from the foothills of the Caucasus regions. That carrots, along with that seeds of queen anise lace, and they have collected the black carrot seeds, they have introduced into the European regions, particularly in the France and Netherlands. And after introduction of these, that the carrots which are belongs to the Afghanistan region or Caucasus regions, these area which is lying in the subtropical regions of continent. If you see the subtropical zones, that whatever the carrots are lying, those carrots are having the higher amount of anthocyanins as well as lycopenes. So lycopenes is nothing but that whatever the nutrients which are there in the carrot, the same amount of nutrients there in the carrot also. Those red carrots and black carrots which are lying in the subtropical zones, that carrots only they have took into European countries along with the yellow carrots also. The yellow carrots and black carrots and red carrots which are there in the subtropical zones, particularly in the Afghanistan region or Caucasus regions and foothills of the Caucasus regions, those carrots which was taken from the subtropical zone to temperate zones, particularly the European continent, they introduced. If you see these the world continent, the tropical zones which that whatever the carrots which are there in the tropical tropical or subtropical zone, those carrots which has the higher amount of bioactive compounds, but these carrots has less amount of water. Because of the less amount of water, these carrots are the higher amount of nutritive value than the European carrots. That whatever the tropical carrots or subtropical carrots are there, it is all higher amount of anthocyanins. Even if you see the morphology of the plants, especially these carrots, they are having the in leaves also as well as the shoulder also, they have the typical dark green leaves, and even the dark patches will be there on the shoulder, particularly in the tropical carrots. You can see here these whatever the zones which are there in the subtropical zones or tropical zone, these zones too, they took into the temperate zones. Once the carrots was introduced from tropical zone, subtropical zones carrot to the European zones, the European zones, once it was introduced, the black carrots and red carrots and yellow carrots which was introduced into European continent, completely the climatic patterns is different. The carrot was introduced into the European country. It is getting a lot of mutations, whatever the carrots which are introduced into European continent, due to the mutations, the single cell mutation. Black carrots which is turned into yellow carrots, the yellow carrots which is turning into orange carrots. But if you see that whatever the carrots which are there in the tropical zones or subtropical zone, these carrots which has a typical the roots are very, very sharp in nature and tail in nature, and these tailed nature are the sharp roots which is turning into the blunt root in the European zones. So that the low amount of water is there in the tropical carrots, but when it was introduced into the European regions, because of the cooler condition of the European conditions, the carrots which has the higher amount of water in the European carrots, because of that, the orange carrots which has the higher amount of water, very tasty, and these carrots was selected by the the royal princes of Dutch as well as France, those people, they have selected the orange carrots, that orange carrot which was distributed completely in the European zones. If you take the tropical zones are how the tropical zones is nothing but it is a warm season crops, or that subtropical zones, that warm season crops also will come, and even cool season crops also will come. But if you take particularly in the European zones, particularly if you see these zones, if you take, this is the Mediterranean zones, this Mediterranean Sea, which has the typical climate compared to the other world climate. Because if you see these the both the side of the tropical zones, which is lying the tropic, the both side of the tropical zone, subtropical zones will be there. Both the side of the subtropical zone, temperate zones will be there. But if you see this is the Mediterranean zones which I am marking. This is the Mediterranean zones. But when this the equator regions too, when you are moving towards the northern words or southern words, the temperature will be the temperature will be less. Means the temperature you will get the sun rays also will get very the less amount, because of this that cool climate which you will get from the north pole or south pole. So normally what will happen, these, if you take the equator regions, both the side of the equator regions, the tropical zones will be there. When you travel from equator to zone to the temperate zones, that see the particularly in the North African regions, here only the Saharan deserts will be there. Above the Saharan deserts, the higher amount of the high amount of heat, as well as the high hot air winds which will blow from the Saharan deserts, which will be go above the Mediterranean Sea. So when this cold, this hot air which is blowing above the regions of Mediterranean regions, which is confluence with the cold air of European conditions, which is getting the typical weather in the the coastal zones of southern zones of European regions. Because of this one, there is many cool season crops like cabbage, cauliflower, broccoli, Brussels sprout, nool cole, radish, these carrots. Typically, Where it was stopped, ma'am? Yeah. Yeah, here. Here. Okay. Am I audible? So you see, if you have seen that cold crops which having the higher amount of glucose inlates, and that that some of the vegetables which does not have any type of colored compounds, but there are some vegetables which have the higher amount of chlorophyll. Those has the higher amount of vitamin A, and some of the vegetables which will turn into some of the vegetables or some of the fruits which will turn into orange or yellow color. Those which will be normally the chlorophyll which will convert into chromophil. So when the chlorophyll which will convert into chromophil, that the higher amount of beta carotene, or it is the higher amount of vitamin C which will start increasing. And if there are some vegetables or some of the fruits which will be turning into red, those which will having producing a lot of lycopenes. And if you take lycopenes, if you take pomelo or red kova, or the ripe raspberries or some of the capsicums, some of the strawberries which has the higher amount of lycopenes. Even if you see take mamordica, even if you take bitter gourd, the seed aerial also which has the higher amount of lycopenes. If you say take some of the vegetables, some of the fruits which has the black colors or different shades of purple and pink, these vegetables having the higher amount of anthocyanins. If you why we are talking about the different compounds, if you take in carrots, the red carrots having the higher amount of lycopene, black carrots having the higher amount of anthocyanins. These lycopene rich carrots are the anthocyanin rich carrots which will be there only in the tropical regions, or so, the tropical regions means that Afghanistan regions, Iran, Iraq, as well as if you take the western part of India, you can find the higher amount of particularly they will talk about kali carrot or kacha carrot, or this is black carrot, which is will be lying in the western part of India, particularly Rajasthan as well as Haryana regions. These normally the black carrots which will be utilized for the animal for feeding purpose, as well as now they have released as a variety in IRI, that is Pusasita. Normally this variety is utilized for the extraction of colors. So already I was mentioning that usually that normally the chloroplast which will convert into chromoplast, the compounds will be enhanced in the carrots. So that why I was mentioning that black carrots which having higher amount of anthocyanins, red carrots which has the lycopene, and orange carrots which has beta carotene and alpha carotene, and yellow carrots which has the higher amount of xanthophylls and luteins, and some carrots which has does not have any type of compounds. So those which does not which has the lower amount of carotenoids, or this nil amount of sometimes what will happen, we on our body we will get when we will get to beta carotene or alpha carotene very higher amount, we will it will get utilized. But if you take parsnip or white carrot, does not have any type of beta carotene and alpha carotene. During the evolution of the carrots, if you see the carrots originally, whatever is there in the Afghanistan region, that the upper portion of the carrots, if you take red carrots or black carrots or yellow carrots, the top portion of the shoulder, or that near to the collar regions, you will see the yellow, typical green color regions. Green color portions will be there in the yellow color regions, as well as black carrots. The top portions you can find the chlorophyll productions, as well as the black carrots also. The top portion of the shoulder, the green color portions will be there. If you take these crops, this is carrot is highly cross-pollinated crop. If you take the two type of crops, the self-pollinated crops or cross-pollinated crop, the cross-pollinated crop doesn't, if suppose why it has cross-pollination, if any plants which has the attractive flowers, or any plants which has the minute flowers or white flowers, or we are not able to we are able to dissect the male and female production male flower easily. Sometimes male and female flower we cannot predict very easily. Those plants which will be causing the higher amount of cross pollination. So now if you take the carrots is compared to other cold crops, other vegetable crops, which is typically producing the different type of flower inflorescence, that is compound ample you will get in this carrots. Normally what will happen, this is I was mentioning that it is a biennial crop. So when you start sowing the carrots, normally what will happen, the carrot seeds have the higher amount of dormancy. Though if you directly, if you seeds sow the seeds of carrot, it does not germinate. So when you have to soak the carrot in the water in the overnight around 12 hours to 24 hours, then again after soaking the seeds, again you take out the seeds from the water and you dry it, again you soak in the water, alternate wetting and drying. If you do that, whatever the dormants are corrupt, whatever the seeds inhibitors which are present in the seeds, that will get removed. Then after you start sowing, the seed will start germinating. Then that if you take the tropical carrots, it will start producing the complete roots 120 days. If you take the European carrots, very short durations, around 60 to 90 days. Before that, you will get the roots. But in case of tropical carrots, it will take 120 days to complete production of the roots. So that after complete production of the roots, the best root you select for the best roots means as how you, as a consumption purpose, what type of roots you will take. By that root we have to select, then we have to take for the seed production purpose. So usually what will happen, that tropical carrots, it is annually, the annual means that does not enter in. I was mentioning that the European regions, whatever the carrots are there, which will be like biennial nature, but whatever the tropical and subtropical regions carrots are there, those will behave like annual carrots. So that once the root production is over, that the annual carrots is directly to start bolting without interrupting the any entrance to the plant systems. But if you take the European carrots, once you select the roots, we have to wait for the seed production in the second year. So what we will do, we start producing the roots for our purpose. So but it will enter into reproducing person in second year only. So for that purpose, what we need to do, once you have to select the roots for our purpose, then we have to make steckling. Steckling is nothing but only the top portion of the roots we have to keep, and the bottom portion of the root we have to cut it. So only the top portion of the roots means that, if you see in this image, only that top portion of the roots and top portion of the shoot, you have to keep it. This is called steckling. The stecklings will be planted. Particularly in the European carrots, you will prepare stecklings. The steckling will be planted. Once the stecklings will be prepared and you planted in during the spring, especially normally what will happen, I was mentioning, all cool season crops are Mediterranean origin. I was mentioning the southern coast of Mediterranean Sea, whatever crops are originated, those crops will behave like a biennial crops. Those crops which will start producing the economic parts only in the cool season crops. Cool season only it will produce the economic parts. So once it will finish the economic parts, the plant will start entering into bolting. Means that plant will start, enough vernalizations or enough cold temperature will be getting in the European continent. So they will enter into flowering in the temperate zones. But when you take in the tropical zones, that is not bold. So what we have to do, what they will do with European people, they will start preparing the stecklings of the carrots. Steckling means once you are disturbing the root productions, means root you are removing the bottom portion. So what will themselves they think that my part of the plant is removed, so I will as early as possible start producing the flowers, then I will finish my reproductive system. So that once the stecklings are planted, the stecklings, after planting the stecklings, the bolting will come out from the roots. Then it will start producing the umbels in the carrot. So where I stopped, I was mentioning that the carrot after 90 days or 120 days, 120 days of carrots in case of tropical carrots, 90 days carrots of European carrots, it will start producing the typical umbel. Umbel means compound umbel. The compound umbel which will be coming out of from the roots, then that umbels which will be used for the crossing purpose as well as even normal seed production purpose also, the umbels will be utilized. So that I was mentioning that we can take into two phases of carrot. One is vegetative phase, another one is reproductive phase. The vegetative phase means up to 90 to 120 days only the economic parts of the root productions will be done. Once the economic part of root once produced, then that roots we will take for the seed productions. So seed productions, if you take in Jammu Kashmir or Leh and Ladakh, we will start we will harvest the roots, we will store the roots in the pits. Then after we will get enough temperature, then normally what will happen, that the temperate regions, that temperature will be very, very low. when the temperature will be normal, like March month, no, those at that time only they will start planting the stecklings. Then plant will start bolting. So the bolting time you have to start crossing and as well as selfing the plant. You can see here, these are the different type of root productions. If you see the 10 days, only the plant will start producing the those two type leaves, and it will start producing only the thin roots which will be coming out. And if you see the different duration time, only the roots will start producing the carotenoids only 60 days. Even I have seen the lot of papers, only the start accumulating alpha carotene or beta carotene or zeta carotene or delta carotene or all type of carotenoids, it will start accumulating only in the 45th day, and it will start completing, or the almost the peak of the carotenoids will be happen in case of European carrots 75th day. But if you take the complete accumulation of lycopene or anthocyanins or lutein which will be there in yellow carrots, that will start accumulating only in 120th day. So if you want to get the higher amount of nutrition in case of tropical carrot, what are the carrots which we are producing in the North India, those carrots are tropical carrots, the plains we are growing. Those carrots only after 150th day we will get the higher amount of nutrition. So that carrots only we have to make for any seed production purpose. After 120th day you have to select it. But in case of European carrots, 90th day you have to select it. See, usually once you start planting the stecklings, that plant will start producing the primary. You can see here in this picture that whatever the once that the flower stalks which will be coming out of from the center portion of the roots, that will produce only the first primary umbel. It will start. Primary umbel means very large umbel. But then in second layer of these flower stalks which will start producing the secondary first umbel, and another one is secondary second umbel. Likewise, the sequentially it will start producing the lot of umbels from the carrots, but only the economically or whatever the umbels we will select for the breeding point of view or quality seeds purpose, only the primary umbel S1 and S2. These two type of umbels which will be utilized for the selfing as well as crossing of the carrots. So normally what will happen, you can see here the different type of umbels are there. This is umbel, right? So normally what will happen, this is compoundable. I was mentioning that this is a small umbels are there. Small umbels are there. These small umbels, how it will start opening the flowers. Only the outer you can closely observe this flower. Only the outer whorl of the flower only which will start producing first. So it is called peripheral nature, means only the outer portion or outer whorl of the flower only which will open first. Same manner, there's all small umbellets which will start producing the flowers. Similarly, after opening of this outer whorl of the outer individual umbellets, the same way these inside the umbels also start producing the flowers. Okay, so this manner only the flower will start opening. So what type of flower will be there? I was mentioning this here. You can't predict the male portion and female portions very easily. Naked eye means by using of lens or minute manner only you can identify the male reproductive organ or female reproductive organ. So usually what will happen in case of cold cross is only protogynous type of flower. Only gynoecium will mature first, followed by male will mature. But in case of carrot, the androecium will mature first. Only the anthers will start producing the mature first, followed by female portions. Then so this protandry type of system also which will induce the highly cross pollination. You can see here I have mentioned that only the outer whorl of the flower will mention. What will happen in case of sir, you told me that primary umbel and secondary first umbel and secondary second umbel only which will start producing the good quality seeds. That why you are saying like this, you can ask questions. So normally what will happen, whatever the early season or early spring season, whatever the flowers are coming out from the carrots, those flowers which will typically you can see the bisexual flower, means the male and female portions will be well developed nature will be there. But in case of lateral part of the umbels, only the predominantly you can see the androecious type of flower. Only androecious means only that male portions only will be there. Female productions will not be well developed. Structures will not be there in the latter part of the umbel. So because of this one, whenever you are doing the crossing or selfing, you have to select only the primary umbel and secondary first umbel or secondary second umbel. So this is the different the maturations and what type of umbels you will select for the selfing and as well as crossing. And if you see here in these flowers, this is typically you can find the androecium or the male portion of the flower, right? And you can see this is the, I was mentioning that the queen anise lace, that the wild carrots, which are wild carrots, that is that does not produce the roots, white roots will be there, very few roots will be there, that will be the lying in only Afghanistan foothills of Caucasus regions. Only those carrots will start producing this type of colors, like pink type of umbels also will be there. Even if you see the carrot field also, you can find some of the parents, some of the roots, some of the parents, some of the carrots lines to start producing pink type of flowers also. And you can see here the two type of umbels. One is the green color umbels also will be there, pink color also. Usually the honeybees is the major pollinator in carrot, and also different type of the involucre also which will be there in the bottom of the umbels. Sometimes that complete developed involucre will be there. Sometimes the involucre is nothing but it is a leafy structures which will be there, and the bottom portion of the carrots. You can see here the bottom portion of the umbels. You can find this involucral bract will be there. This is if you take Jammu Kashmir or Leh and Ladakh regions, under protected conditions, especially insect fruit house or plastic polyhouse, they used to plant the stecklings. Then the plant will start producing the like these floral parts. Means many many the umbels will come. We will under the protected conditions only they will start producing the means we will do the selfing or inbreeds, or we will do the crossing. Everything we will do inside the polyhouse. Usually what will happen, this is a completely umbel. You can see here the five petals will be there, right? Five petals will be there, five sepals will be there, and five the androeciums will be there. Androecium also five androeciums, and the stigma will be two bistigma will be there. You can see here two stigma regions will be there, and the two stigma when you get the successful pollination and fertilization, so that successful pollination and fertilization will turn into two types of mericarp. So when you crush the seeds of carrot, if you take coriander or anything, so it is typical. Not only single seeds will be there, it is here two seeds will be there. So that when you crush the seeds of the complete developed seeds of carrot, you will get two seeds. So two mericarp you will get it. So the two mericarp which will turn into the complete seeds. So usually that it is a normally what will happen, normal conditions, the flowers are fertile, androecium and androecium gynoecium well developed will be there. In sometimes what will happen, the sterility which is happening because of the high temperature, because of some mutations which is happening in the cytoplasmic regions, they have the two type of flowers. One is cytoplasmic male sterility. They cytoplasmic male sterility they have identified because of the cytoplasmic regions. Cytoplasmic regions means the outer side of the nuclear regions, which will cause some of the genes will not produced for the reproductive purpose. Because of that, what will happen, that androeciums or the androecium part will not produced perfectly, as well as what will happen, instead of androecium some other floral like structures which will be formed instead of androecium. So this type of morphological changes or morphological mutations which will happen in the flowers, that will be used for the breeding purposes. So two type of three types also. Now recently they identified five type of male sterility also, but two types commonly you can find it. One is brown anther type of male sterility, another one is petalite type of male sterility. You can see here that it is the brown color the male sterile parts are formed. This is a brown anther male sterility, and instead of anthers, that leafy like structures, it is called petalite type of male sterility. So two type of male sterility, it is petalite type of male sterility. Petalite type of male sterility, which is commonly utilized for the hybrid seed production. So you can see here, this is the how the morphological features which will be there in the sterile portion, sterile flowers and fertile flowers. You can see here the fertile flowers, the typical androeciums and the anther portions which will be there. But the sterile flowers, you can't find any type of these type of parts. So you can see here also the closed view of sterile and fertile flowers. Fertile flowers, this is, you can see, completely white, and that androecium, gynoecium part you can find it, but here it will not be there. And by using of these systems, they have developed some of the hybrids also. This is the different shape of umbels. Normally what will happen, if suppose you want to produce the hybrid seeds, so generally what they will do, 3 lines of sterile plants or 4 rows of sterile plants they will plant. Then afterwards the 2 rows of fertile plants of carrots they'll keep. Then they will start producing the hybrids by using of these systems. I was mentioning that different type of how they will produce the seed productions. I was telling that once you select the roots of economic parts, you prepare the stecklings, and stecklings will be planted under the greenhouse or polyhouse or insect-proof net house. Once you plant the stecklings by the spacing of 30 centimeter in 90 centimeter spacing, then that after stecklings planted, then it will start producing the floral stalks which is coming out. That flower stalks is coming out from under the green insect proof net house, you can see. Then it will start producing the umbels. Then umbels, which after successful selfing, you can start get the fruits of the schizocarps. So that after during the flower fruit productions, we can do the different type of roguing they will do. Sometimes it will start from the root productions. Sometimes it will start from the seed production. But what they will do, if suppose you want to produce the only one varieties, you should not keep any varieties. You should not get any mixes inside the field. So different stages you have to go for the roguing, that is pre-uprooting, after uprooting, or bolting time, and flowering stages. Flowering stages also, if you see any floral variations are there in the umbels, if suppose any floral variations there, that you will be discarded. So here 2 type of seed production method, only seed to seed method, root to seed method. I was mentioning that in case of Leh and Ladakh, first they will after harvesting the roots, they will make stecklings. They will be stored inside the pits. Then once you get the congenial weather, then they will go for the planting. See what I have done in the my experimental purpose, and I was selected that 4 lines, that is IPC 122, that is a red color carrots, that is Pusa Rudira, and Pusa Meghali, that is a orange carrot, IPC 126, that is a black carrot, and white pale, that is a Pusa Gulfi, that is a yellow carrots. The 4 carrots I have taken. You can see the different morphological features of the flowers also. The black carrots, you can see the purple type of flowers. White pale, you can find the pure milky white type of flowers colors you can find. And remaining red color flowers, that remaining small red patches will be there on the upper surface of the umbels. These are the different morphological features. So these flowers, what I have done, how you have to do the selfing and crossing. I was mentioning it is only the outer portion of the flower only will open first. So you have to emasculate only the outer portion of the flower first. Then whatever the central portion of the flowers are there, central small flowers are there, that you have to remove it. Then whatever the plants you want to do as a male parents, you bring that male parents and you start crossing in this. After successful crossing, you will get the fruits from this. This is the different stage of flowering and different stages of how you will select for the hybridization purpose. You see, sir, I have selected matured, well matured flowers. You can see here third flower and fourth flower. Only the outer floral of the flower only is start maturing. Here you can select only outer flower of the flower for the emasculation purpose. After emasculating, then you can see here the 9th flower. Complete central portions are removed. Only the outer floral of the flower only I kept. Then we can start crossing by using of any already this is highly cross-pollinated crops. If you select any red color or white color or black color, that flowers, before opening of the flowers, you have to cover it. From there, you collect the pollens and you do crossing in the selected plants of female parent. So you can see here that I started producing the male based hybrids. This is the how we will produce the inbreeds under the shade net, insect fruit net house, and this is the open plot how we will do the selfing. These are the different stages of the crossing systems. And you can see here that I was mentioning that how the different colorful carrots was evolved. He is the man who is working for the carrots. He spent his lifetime, Dr. Philip Simon. He has done enormous work on the carrot, particularly genetics and breeding. He has done enormous work. Without him, there is no morphological diversity will be there in USA or European carrots, whatever. The temperate carrots work. The temperate carrots means only that orange carrots work was done by him, that is Philip Simon. Usually that carrot was, evolved from 900 years ago. And you can see here that that center portion I was seeing that minute pink color discolorations will be there, right? You can see here outer, the complete umbels, only the center whorl of the flower. This is a non-reproductive part. This is non-functional to the umbels, but this will happen only in the queen anise lace. That is QAL. That is QAL or queen anise lace they used to call. This QAL special features will be there in some of the carrots. So we can identify that what type of floral structures does not have any anthers or gynoecium part. Only that floral like petal-like structures will be there that does not have any functions. So these functions is coming from the Q in lace. These are the, you can see here that what are the, I was mentioning that for reproductive purpose only, reproductive enhancement purpose, they have came from the Europeans and they have collected seeds of carrots. That carrot is very small in nature. That is, you can see here, like spiny like structures will be there, that wild carrots. You can see here this Afghanistan in India, that first domesticated 900 AD only the carrot was evolved. That is, you can see black carrot and yellow carrot. That is, it came to here. And you can see here, these are the different colors which was evolved from the single black color. Why I am talking about the different colors? See, when you talk about the black color, red color, or orange color, yellow color, if you take, I was mentioning about breeding program. If you take black color as a female parent, and if you start crossing orange and red and yellow, only the black will dominate on the outer side of the skin. If you see the carrots, the carrot has the different type of, you see when you cut the carrot, see when you cut the carrots, what will happen? That outer side, that is a epiderm layer. Epiderm layer, there is one color. You can see here. If you see, this is epiderm region. Epiderm region is nothing but the skin of the carrot. If you see the center core of the region, that is called xylem. And in between the outer side of the skin as well as inside the xylem, in between these regions called phloem regions. Normally what will happen, if you take whatever the carrots which are there in the European regions, the xylem regions, phloem regions, epiderm regions, all colors will be same. There is no color differentiations will be there in European carrots. But if you take in the Afghanistan Indian carrots, that if you take, you can easily fabricate the xylem and phloem and epiderm regions. These regions, 3 colors variations you can find in case of red carrots and black carrots. So what happened? These, whatever the work they have done in particularly in the European regions only they have used orange carrots. But what I have done it, black carrot I have taken as a female parent, and black into red and black into orange and black into yellow and black into this. So like this manner I started crossing. In reverse manner also I started crossing. So whenever you are using black as a male or female parent, only the black will dominate only in the epiderm region as well as the center core of the regions. When you bite the carrots, the center portion of the carrots you will feel very, very hardy, means very, very hard in nature. But when you eat the carrot, the center portion should not be hard. Always we, as a carrot breeder, we used to select the coreless coreless carrot cultivars or coreless carrot only we will target. But the coreless carrots, the coreless means the core regions, that is the central core regions, but that also will express as a black color. So when I am using asita, if you take pusa asita, it is a black carrots. If I am using as a male parent or female parent, the outer portion of the root, that is a epiterm region as well as the central core regions, only the anthocyanin will express as a dominant. Okay, that if you take even red color also, black into red and black into orange, everything will, black will express in the xylem region and black will express in the epiterm regions. What will happen in case of phloem regions? Whatever the light colors you are using as a male parent, if suppose black into yellow you are using, the yellow color will express in the phloem regions. If suppose red color you are using as a male parent, the red color will express in the phloem regions. Suppose red, yellow, orange, this color only whenever you are using as a male parent, that will express in the phloem. So what I want to say that the black color will dominate, dominant in only in case of skin, particularly in the epiterm regions. So purple is dominant over red and red is dominant over orange, that orange is dominant over yellow. This is the finding I have studied after 4 years of my PhD. I have concluded that the purple is dominant over red and red is dominant over orange and orange is dominant over yellow. This gene expression will happen only in the epiterm layer, but that will be happen in case of xylem also. But in reverse will happen only the light colors like lutein, yellow dominant over orange and orange is dominant over red and red is dominant over black. This type of genetic pattern you can find only the phloem regions of the carrots. So by this manner, if you see the whatever the European carrots, you can find the different shape of the roots. You get Paris market or Oxford, Nandis, Sandini, Tanvers, Imperator, Croda. These all carrots has the typical shape. Even if you see the bottom portion of the carrots, this is very, very blend in nature. But whatever what is lacking in our Indian carrots or Afghanistan carrot, the roots are very, very sharp in nature. That is not in blend in nature. And also just mentioning that our carrots are highly nutritional value, but the orange carrots are high amount of water. Because of that, juiciness is there. Because of that, is very, very popular. If you see here the different type of carrots, what you can find, if you take European regions, that long orange group and horn group. Horn group means very typical small shaped carrots, that is horn groups. But long orange groups which has more than 90 to 120 or 160 180 centimeter length carrots also you can find only in the European regions. That Europe regions, that whatever the horn group carrots are there, long orange groups are there, which was introduced into the Europe, particularly North America. After introduction of this horn group and long orange group, they crossed into long orange group and horn group. They developed imperator carrots. Imperator carrots means those carrots which is suitable for the bunching purpose. If you go to the market, only the carrot bunches are available. These carrots are suitable for the bunching carrots. Even mechanical harvesting they used to do. Even it is very useful for the it is like you might be heard about baby carrots. The baby carrots is it is a the small alteration they will do in the outer portion of the carrots, and they will make into small cutlets of the carrots. That is a small baby carrots, very, very popular by using of this imperator, in particularly USA. This imperator was very, very popular in America by using of the horn group or orange group, long orange group. But if you take the Afghanistan regions, which was lying the black carrots, red carrots and yellow carrots, this was not utilized properly. So that we have to utilize properly by producing the nutrient-rich carrots. I was mentioning that each layer, that outer portion, epiterm regions or xylem regions or phloem region, this 3 layer, I want to bring this all nutrients together. So we can get the rainbow carrots or multi-nutritions or multi-minerals packed carrots we want to produce. So for that purpose, I started crossing these all parents into black into yellow and red into orange and orange into yellow. By this way, I developed individual, means A into B and B into C and C into D and D into E. So all possible combinations I did crossing, and I derived F1. Then all F1 again I crossed into, brought it into single F1, so that all genes or all colors which will be presenting in the F1, so that afterwards start producing the selfing of those parents. So you will get all type of colors in the carrots. See, whatever the carrots, if Japan, if you go to Japan, they have developed a heat tolerant variety, that is Koroda. Koroda scientist, he is a scientist who worked for the heat tolerant variety. Koroda he released. The Koroda was introduced into South America, and they started, that non-conventional regions, they started producing the carrots in the South America, particularly Brazil regions, and those carrots was Koroda carrots was crossed with the Imperator, and they brought first root knot nematode variety, that is Brasilia. So what we have done it in Indian regions, we started producing these all tropical carrots. He is the man who started producing the baby carrots. So usually what will happen, that whatever the we want to produce that black carrots into normal carrots. What is the importance of this black carrots? The black carrots which has the water soluble pigments like cyanidin 3-glucoside, cyanidin 3 galactosides. So normally what will happen, I was mentioning that when you harvest the black carrots, that after, what will happen, when you harvest, the color will be very, very attractive. But when after due course of time, the color will start degrading. But what will happen in case of bioavailability of the compounds, especially the cyanidin compounds and cyanidin compounds, which will start accumulating due course of time. So you just put in water, that water will get again that water will get absorbed by roots, that all nutrients will get activated. So this is the new means what we can say, that it is the attractive colors which will be come out from these carrots, as well as the nutrition's bioavailability also is very, very high. And if you see, these are the different lines which are developed from USA. But what we have done it, you see here that I have made into black into yellow, and black into red, and black into orange. And in F1 we have got only the black. In the F1 we got, when you are crossing in black into red, only red will express say dominant in F1. In red into yellow you are crossing, only yellow will express in F1. But in case of F2, you can see that a lot of diversification is there. By these studies, we identified that I was mentioning, that when you are crossing into black and yellow, only yellow will express as a dominant in the epidermal layer, and purple will express inside the core region. So likewise, whatever the colors are there, we have graded, we have grouped into RHS color chart. You can see here, these are the different core colors expressions in yellow carrots, and the different variations in the black carrots, and different variations in the red carrots. You can see these are the different color variations inside the black carrots. These are the different color combinations of this is not just color. When we are talking about the carrots, tropical carrots, you, instead of talking about yield, you talk about the nutrition. So this much kg of anthocyanins you'll get from the sector, 100 kg of anthocyanins you'll get from this per acre. So by this way, if you talk, it is a meaningful, as well as if you are getting higher yield, that is not mean. Along with the higher yield, we should get the higher amount of nutrition also. By using of the studies, I have published a lot of papers also. If you see here, by using, by these studies, I come to know that whenever you are using the black carrots, which will express, and which we can use as a parent for the heterosis breeding program. So by using of the different type of studies, I know that I was mentioning that purple is dominant over. These are the different type of gene expression studies in case of epiderms and phloem and cortex regions also. And if you see here, these are the different populations we have done. And see here, white into black, and the black, I have raised the F1, and F1 was self, and F2 population I got. In F2 populations we got different variations. The F2 populations was grouped into different by using of different colors. What will happen in case of carrots, you will get 4 groups in F2 populations, like completely black, and top portion will be black, and the bottom portions will be light red, and sometimes completely light pink color will be there, and sometimes the parental types also will be there. What will happen in case of parental types? The parental types you won't get any type of nutrition. But when you get the combinations of top portion black and bottom portion red or light pink colors, these having the higher amount of nutrition as well as the marketable attractive values which has. Usually what will happen, the lutein is mainly it is useful for the macular degeneration diseases that happen in the eyes. For eye purpose, that yellow colors is most important. So we want to bring this lutein content inside the carrots of inside the black carrots. So already that black carrots has higher amount of anthocyanins. So along with the anthocyanin, normally what will happen, the black carrots which has the higher amount of anthocyanin, because of this one, the black carrots you don't, while eating you won't feel that any you will feel somewhat astringent taste. But when you are adding the yellow carrots inside the black carrots, what will happen, the taste of the carrot is exactly carrot, but the astringent taste will be very less. So what will happen in case of black carrots, the black carrots, because of the higher amount of anthocyanins, that anthocyanins normally it will mask the all sweet taste of the carrots. Usually what will happen, normal carrots what we are eating, black orange carrot and red carrots, the normally the glutamic acids will be very very high. Glutamic acids, normally this glutamate is a sweetening agents which will be added in the cakes. The same type of compounds will be there in case of red carrots and orange carrots, but these compounds which we can bring naturally from the yellow carrots, that yellow carrots is very very nutritional, so that yellow nutritions we can bring into inside the black carrots. You can see here, so we have developed the population mapping populations around 500 plants. These 500 plants we started screening through different primers, so that we identified that some of the primers which was linked with the purple dominant genes, these are the different populations. And we have identified some of the 7 primers which was giving very good diversification in the populations. By using of these screening, we identified that some of the primers, that is GSSR 134, this primer is very closely linked with the purple genes. So these primers we have utilized for the selection of other carrots also. So through marker assessed selections, we can select that the purple anthocyanin enriched carrot in the F2 means any generations, we can select the best roots. And you can see here we have, I was mentioning that the different groups of carrots like black carrots, and the top portion will be black and the bottom portion will be light pink and light pink color carrots. So if you see the different profiling of the anthocyanins also, you might be seeing that anthocyanin is nothing but not single compound, it is more than 500 compounds. So out of the 500 compounds, the 7 compounds which are very predominantly which is existing in the carrots. But if you see, I was mentioning here that 1, 2, 3, 4, 5, 6, 7, that is a cyanidin compounds or cyanin compounds, this will be very very dominant in case of black carrots. But if you take in light pink color carrots or another top portion black and bottom portion will be red, these carrots cyanidin will be more, not cyanin will be more. Now black carrots cyanin will be very very high, but top black and bottom pink color which has cyanidin will be very very dominant. But in case of light pink color, if you see that RHS 77B in these carrots, you can find that there is a new compounds, that is peonidine compounds. The peonidine compounds which will not present in the other carrots, but peonidine compounds will be there in these carrots, which will be useful for the that nutrition purpose. These are the different you can see here the different lines which are identified from South America. These are the different more than 32 genes which is responsible for carotenoids. This one they have done from European carrots, but we have not done. But we can target for our population. So for our purpose only, by using of these by using of this indigenous collections and indigenous germplasms, we have developed some of the Pusa Gulfi, Pusa Meghali, Pusa Rudra, Pusa Sudha, Pusa Vristi, Pusa Asita. These are the lines which are explored from our indigenous germplasm. We have selected and we made crossing, and sometimes we made by using of CMS system also we have developed some of the hybrids. But what I have done it, that I have, by using of these all parents that I have developed, you can see here all colors which are expressed in the magic population. I was mentioning that all parental like black, red, orange, yellow, these all colors I brought into single parent. So single genotypes, so that you can see that single genotypes you will get all type of nutritions. When you eat the carrots, you should get all nutritions. So that you can see here there are different lines which I have developed. You can see the different color stacks here. If you see if you see that the outer epiderm regions, that black different color, black also is the different colors. I was mentioning that out of 6 compounds, that cyanidine, cyanine, and thionidine, these compounds different concentrations which will be there in the black combinations. Even lycopene, you can see that the yellow variations will be there in some carrots, light red variations will be there in some carrots. Even red also, red color variations will be higher proportions to lower proportions will be there. Wherever the higher proportions of red is there, there is higher amount of lycopene. Wherever the higher proportions of black is there, there is higher amount of anthocyanins. Wherever the higher amount of yellow colors are there, it means that the higher amount of luteins are there. You can see here, these are the carrots which you can utilize for the color extraction purpose, juice extraction purpose, even a normal consumption purpose also. I was mentioning coreless cultivars, but these, whenever you are eating these carrots, you won't feel any hardy nature inside the carrots. Even these carrots, we can make very good carrots shred from these colored carrots. You can see here different carrot slices and the shred also we have made from these original carrots, and we have grouped into different color groups also. These are the lines which are developed from the match populations. You can see purple yellow variations and purple pink variations, and you can see here the different purple and orange variations, and orange types, and purple and red variations. These are the different lines which we are in the pipeline to release. These are the lines which is composed with lycopene, lutein, carotenoids. So these are the promising lines which are derived from the magic population. These are the carrot important lines. These are the different lines which were showing the different type of cyanidine and cyanidine compounds. You can see here that what I have done it, that whatever the carrots are there, that almost whatever the lines which we have developed, those lines which are having the maximum amount of carotenoids, which has around 594 milligram per 100 gram. In case of minimum amount of carotenoids, which has 101.20 milligram. The same amount of anthocyanins also, you can see here that lycopene also very the higher amount, even the carotenoids also very higher amount which will be there. These are the some of the carrots. You can see whatever the carrot lines which I have shown, this is very promising lines compared to exotic lines. You can see here, this is Uzbekistan, completely Middle East countries, Uzbekistan. These carrots are very blunt in nature. You can see this is a round carrots, very small carrots. This is a pink baby carrots like Rudira, and purple cosmic carrots is exactly look like Asita carrots. This is a Persian carrots. This is Nandis types, but this is an Indian carrots which was from the all our European continent. They have developed these carrots. This is a carotenoid-rich carrot, that is carotyl carrots. This is Chanthani, white Chanthani. It is exactly look like parsnip, but this is the carrot which are very popular in the European regions. This purple Chanthani, that is the shape of the carrot is exactly look like triangle shaped carrots or conical shaped carrots. This is all other rainbow carrots of black nebula. This is a high source of anthocyanins, and this baby yellow punched carrots, gluten-rich carrots, that is baby white punched carrots, and these are the other carrots lines. These are the some of the materials which we brought by using of petalite type of breeding program. We have done it. And this is the Pusa Meghali carrot. This is a rich source of whatever you have seen the colorful carrots. Those colorful carrots parents which I used, that is Pusa Meghali, the higher amount of rich source of beta-carotene. Rutira, which has the higher amount of lycopene. This is also one of the parent which I used. And Pusa Gulfi, this is yellow carrot. This is also one of the parent. Pusa Sita, that is anthocyanins which we used. You can see here, these are the combinations of different nutra-rich lines like IPC 2616, IPC 11226. These are the lines, these lines which we can use for the future breeding program. Thank you.
First, if you have any questions, please ask.
Good afternoon, sir. This
Yeah,
is Naganjali from Professor Jaishankar, Telangana Agriculture University.
yeah.
Very